This content is not designed for use as a medical reference to diagnose, treat, or prevent medical illnesses or trauma. If you have questions about the diagnosis, treatment, or prevention of a medical condition or illness, please consult your personal physician.
Welcome back, everyone. In today's lecture, we're going to take a closer look at a language-based learning disability known as developmental dyslexia, which affects about 5 to 10% of school-age children and tends to occur more frequently in boys than in girls. Before we discuss the disorder in detail, though, let me introduce you to Brian, who has dyslexia. Brian is currently an adult, but he was kind enough to share his childhood experiences with me. When Brian was in the 4th grade, he exhibited certain problem behaviors. For example, he refused to read out loud when asked by his teachers. And when he was given a verbal list of tasks to complete, he would either not complete them at all or complete them out of order. Though he read at home, he mainly stuck to comic books or books intended for much younger children. And even then, his parents said it was really hard getting him to read out loud. When he did, he was slow and often had to read the same sentence 3 or 4 times before moving on to the next one. Brian told me that he refused to read out loud because he felt embarrassed and stupid. He noticed that other kids read out loud much better than he did, and that they were reading harder books. His teachers complained that Brian would often read questions too fast or not at all, and would often get answers wrong unless someone else read the questions to him. They thought Brian was smart and concluded that he was just lazy and was refusing to work as hard as the other kids. But Brian's parents disagreed. They stated that when he was doing homework that relied less heavily on reading, like math, he spent a lot of time studying, and he did well. However, when he tried to do assignments that required large amounts of reading or even spelling, he didn't do well at all. His parents also acknowledged that he'd have trouble reading ever since he started learning. After a back and forth with Brian's parents, the school system finally gave in and agreed to have Brian undergo neuropsychological testing. The doctor concluded that Brian had an above-average IQ, but also significant issues with verbal learning, including reading, which was consistent with a diagnosis of dyslexia. He confirmed that the issues Brian was having in school matched the neuropsychological test findings. To address these issues, the doctor recommended that Brian use resources consistent with something called the Orton-Gillingham approach, which was the first teaching approach specifically designed to help children who struggle with reading. It is a direct, explicit, multisensory, structured, and diagnostic way to teach literacy to children who struggle with reading, writing, and spelling. The doctor also explained that dyslexia is not a curable condition, but that it can be managed. He said Brian would be able to learn to read effectively, but it would be challenging for him to become a great reader or writer. He re-emphasized the fact that Brian was not stupid, noting that people with dyslexia tend to just think differently, and that Brian could possibly use this to his advantage later in life. As I mentioned earlier, Brian is an adult now, and a very successful one at that. He graduated from college with a bachelor's degree and is currently employed. His story helped me to understand some of the struggles that children might go through who have this condition. So with Brian's story in mind, let's move on to discussing some of the signs of developmental dyslexia, how it's manifested, causes of the disorder, and the best ways to help people, especially children, who have dyslexia. Developmental dyslexia begins in early childhood. This is different from acquired dyslexia, which occurs as a result of neurological damage, such as due to a stroke or head injury. In today's lecture, we'll be focusing on developmental dyslexia, which I'll refer to as dyslexia from now on. It's important to emphasize that dyslexia is not due to a lack of either intelligence or the desire to learn. With appropriate teaching methods, individuals with dyslexia can learn successfully. Individuals who have dyslexia typically experience difficulty reading, which directly impacts their school performance and everyday activities. And because reading is a basic skill that directly relates to most school subjects, children with dyslexia are at a disadvantage in most classes and may have trouble keeping up with peers. Dyslexia can also lead to social problems. If those who have it don't receive treatment, they can develop low self-esteem, anxiety, aggression, and behavior problems, as well as becoming withdrawn from others. Moreover, issues can carry over into adulthood, since those who experience difficulty reading and digesting information in childhood may not reach their full potential. This can have long-term educational, social, and economic consequences. Luckily, this was not the case for Brian, who received the intervention he needed. Children who have dyslexia have a heightened risk of developing attention deficit hyperactivity disorder, so ADHD, and children with ADHD have a higher risk of dyslexia. Dyslexia also frequently co-occurs with specific language impairment, which is a communication disorder that interferes with the development of language skills in children who have no hearing loss or intellectual disabilities. Some signs that a child might be at risk for dyslexia include reading below grade level, avoidance of reading, trouble answering questions, difficulty remembering the sequence of things, a hard time perceiving similarities and differences in letters and words, an inability to sound out unfamiliar words, spelling difficulties, and taking a long time to read or write. When you think about it, reading is a very complex and integrative neurocognitive process. It requires the precise visual recognition of letters and letter combinations and the conversion of this information into appropriate sounds using what's known as primary grapheme-phoneme mapping. A phoneme is the smallest unit of sound, and a grapheme is a way of writing down a phoneme. This process therefore requires the acquisition of accurate visual and auditory information, knowledge and memory of phonemes, the use of selective and sustained attentional processes, and executive functions. In other words, it's a multifactorial process that involves multiple brain areas and systems. For this reason, dyslexia is very heterogeneous, with different individuals exhibiting different symptoms. Researchers have attempted to classify dyslexic individuals according to various theories, but different researchers use different classification schemes, and individuals don't appear to fit neatly into one category or another. Instead, they show symptoms that fall under multiple categories. The research findings for the various theories regarding dyslexia are also inconclusive. Given these caveats, I would like to briefly introduce you to some of the most prominent theories about deficits related to dyslexia. We'll start with the most popular theory, known as the phonological theory. According to this theory, dyslexic individuals have impairments related to the representation, storage, or retrieval of speech sounds or phonemes. Learning to read a written alphabetic system requires learning the correspondence between letters and fundamental sounds of speech. If these sounds are poorly represented, stored, or retrieved, the learning of grapheme-phoneme correspondence, which is how we read, will be affected. Phonological theory holds that people with dyslexia have deficits related to phonological fluency, so the ability to generate words starting with a specific letter or in a given category over a certain amount of time, phonological awareness, so difficulty with analyzing, blending, and manipulating the sound structure of words, and verbal short-term memory, so problems with the serial recall of words, naming, and repetition of pseudo-words and sentences. These noted deficits lead to poor grapheme to phoneme conversion, which in turn leads to poor reading ability. At the biological level, this theory is associated with abnormalities observed in the left perisylvian region of the brain, the left temporoparietal region, and the left frontal lobe. A second theory, known as the visual magnocellular theory, states that the underlying cause of dyslexia is not a language deficit per se, but rather an impairment in the visual system, and specifically in the magnocellular pathway. We actually talked about this pathway when we explored idiopathic intellectual disabilities and Down syndrome. In that lecture, I mentioned that there are two primary visual pathways that travel from the eye to the primary visual cortex in the brain. One pathway, the parvocellular pathway, primarily carries information about the color and details of an object. A second pathway, the magnocellular pathway, primarily carries information about motion perception, binocular depth perception, and the brightness contrast of objects. The magnocellular pathway plays an important role in reading. For one, it helps to stabilize printed images. Individuals with dyslexia may have an unsteady binocular fixation, so they may complain that letters seem to move around on the page. It also affects vergence movements. Our eyes are not parallel, but rather converge at about 10 degrees toward each other. For individuals with dyslexia, these letters and words appear to split and move over each other, making it difficult to decide what order they should be in. For obvious reasons, this could affect reading. The magnocellular pathway also may affect orthographic knowledge, which is knowledge of the written form of thousands of words. This knowledge helps us to recognize words in an instant instead of having to decode them, which in turn helps us to read fluently. Finally, the pathway may affect speech sounds that result from this impaired orthographic knowledge. Biologically, researchers have found that the magnocellular cells in the lateral geniculate nucleus in the thalamus are significantly smaller and more disorganized in individuals with dyslexia compared to control participants. A third theory is the rapid auditory processing theory. This theory holds that people with dyslexia have an impairment in the auditory pathway that helps process fast auditory information. Deficits can be seen in the ability to make rapid sound changes and observations of slow auditory transitions. These deficits could easily affect one's ability to read. Biologically, the larger neurons in the medial geniculate nucleus in the thalamus, which have rapid temporal processing capabilities, are smaller in dyslexic individuals compared to control participants. The last theory we'll talk about here is the cerebellar theory, which holds that deficits are related to impaired function in the cerebellum. The cerebellum helps us to automatize overlearned abilities like typing and reading. It also plays a role in motor control and therefore speech articulation. These deficits may lead to problems with converting graphemes to phonemes, word recognition, verbal working memory, and awareness of how letters are associated with particular sounds. Articulation problems from cerebellar damage can also directly affect phonological awareness, which in turn affects reading. Anatomically, individuals with dyslexia have more large cerebellar neurons and fewer small ones than control participants. Research by Gavin Reed and colleagues in 2006 examined the cognitive profiles of dyslexic individuals across domains corresponding to the phonological deficit theory, the visual magnocellular deficit theory, and the cerebellar deficit theory. When these theories were examined as a group, the researchers found that individuals showed impairment on all tests that examined literacy, except for those that assessed reading comprehension. Reed and colleagues found that dyslexic individuals could compensate for their reading deficit by relying on context. These individuals also showed impairment on most of the phonological tests, but not on the visual magnocellular or cerebellar tests. When the researchers examined individual profiles, though, complex and heterogeneous cognitive profiles emerged. In their sample, they found that 86.7% of dyslexic subjects showed a phonological deficit, 13.3% showed a visual magnocellular deficit, and 26.7% showed a cerebellar deficit. Keep in mind that different studies do report different findings. A lot depends on the assessments used in the study and whether deficits are categorized as phonological, cerebellar, and so on. Moreover, some studies may not even measure certain reported difficulties. It is also worth noting that scientists dispute the existence of certain types of deficits, like those seen in the cerebellum. Even when they agree that deficits exist, like those in the magnocellular visual system, there is debate as to whether the deficits cause a reading disability or whether, in fact, the reading disability causes the deficits. So a lot of questions remain to be answered as to whether these deficits exist, how they relate to one another, and whether or not they contribute to the reading deficits observed in dyslexia. Just something to keep in mind if you delve further into this research and its implications. Now that we've talked about some of the proposed classifications of dyslexia, let's examine what scientists believe some of the causes to be. As with the other disorders discussed in this course, dyslexia is believed to be caused by the interaction of multiple genetic and environmental factors. Let's start with the genetic factors. Research suggests that at least 9 to 10 different genes might play a role in the development of dyslexia. These genes have been linked to chromosomes 1, 2, 3, 6, 11, 13, 15, and 18. And a 2017 literature review by Natalia Becker and colleagues suggests that chromosomes 7, 10, 12, and 21 might also be implicated. Twin studies suggest that the concordance rate for dyslexia in identical twins is 68%, whereas in fraternal twins, it is 38%. This signifies that dyslexia does, in fact, have a strong genetic component to it, though environmental factors also play a crucial role in its expression. Different researchers have provided evidence both for and against particular genes and their role in the development of dyslexia. Given the amount of individual variability present in this disorder, it's difficult to make generalized statements about particular genes. Let's say a researcher finds that gene A, for example, can be linked to dyslexia in one person, but gene B can be linked to dyslexia in another person. Are both genes responsible for dyslexia? How is the expression of dyslexia in person A different from the expression in person B? Are other genes involved as well? Unfortunately, we have many more questions than we do answers at this point, and particular genetic markers are not found consistently across different individuals with dyslexia. 4 main candidate genes have been proposed as contributing to dyslexia based on genetic mapping approaches and statistical analyses. A review of these genes appears in a 2007 paper by Silvia Paracchini and colleagues. They are DYX1C1, located on chromosome 15, ROBO1, located on chromosome 3, and genes KIAA0319 and DCDC2, both located on chromosome 6. Although the research on these specific genes is mixed, all 4 contribute to the development of the brain. Namely, they seem to impact pathways involved in axon growth and neuronal migration. It is through neuronal migration that neurons, during fetal development, travel to the places in the brain where they belong. Once they reach their destination, they extend axons and dendrites to communicate with other neurons. Interruptions to this process can result in a variety of physical and mental deficiencies. Some researchers have hypothesized that dyslexia is a result of abnormal neuronal migration. As to environmental factors, those that have been shown to influence the development of dyslexia include premature birth, low birth weight, in utero exposure to nicotine, drugs, alcohol, or infection, and growing up in poverty or with little exposure to literacy-related activity. Now that we've explored what dyslexia is and some possible causes, let's take a closer look at some interventions that can help those affected better manage the disorder. With a proper diagnosis, appropriate instruction, hard work, and support from family, teachers, and friends, individuals with dyslexia can succeed in school and later as working adults. First, it's important to outline the criteria for successful interventions. In the case of dyslexia, scientists generally agree that the intervention should use well-grounded theory to target impaired processes, be evidence-based, be well-structured, systematic, and multisensory, incorporate direct teaching, learning, and time for consolidating information, be frequently revised to address the individual's specific learning difficulties, include training in letter sounds, phoneme awareness, and linking letters and phonemes through age-level appropriate reading and writing, be implemented as early as possible, and be adaptable, as not everyone will respond to the intervention. In fact, a better understanding of these non-responders will aid in adapting an intervention to suit the affected individual's needs. Additionally, in dyslexic people with comprehension problems, interventions should include training in oral language skills, especially vocabulary training. A 2008 study by Claudine Boyer-Crane and colleagues examined 4 to 5-year-olds who were at risk of developing literacy difficulties. Although they didn't include a control group in their study, which is an obvious drawback, they did compare 2 intervention strategies that targeted different skills in these at-risk children. The first intervention was the Phonology with Reading program, or P+R, which targets the development of phoneme awareness, letter sound knowledge, and reading. The second intervention was the Oral Language Program, or OL, which is designed to improve spoken language skills that can in turn contribute to the development of reading fluency. The 4 key elements of the OL program are vocabulary training, narrative work, independent speaking, and listening skills. The children in the study received 1 highly structured intervention by a trained professional each day for 20 weeks. So what did the results show? Well, children in the P+R group did significantly better than children in the OL group on tests of phoneme awareness, letter sound knowledge, reading, and spelling skills. At the same time, children in the OL group did significantly better on tests of vocabulary and grammar. This makes perfect sense, right? The children improved on the specific skills targeted for intervention. And these gains in performance were still being maintained 6 months after the interventions had stopped. The interventions even lifted some children out of the at-risk category of literacy difficulties and into the typical range of reading and language skills for their age. By the end of the interventions, 68.1% of the OL group remained at risk, but only 50% of the P plus R group remained at risk, showing that the P plus R intervention perhaps targeted deficient skills better than the OL intervention. So clearly, science-based interventions can have a beneficial effect. But what about products that claim to improve reading performance in individuals with dyslexia? Do they work? Let's take a moment to talk about one of the more popular ones, colored filters. These include colored lenses for eyeglasses and colored overlays placed over text. Though the use of these colored filters is pretty controversial, they continue to be used by both educators and parents to help alleviate the symptoms of dyslexia, and some individuals with dyslexia claim that they're effective. The research, however, is another matter. Most scientists claim that colored filters are not an effective treatment for dyslexia. In fact, a 2016 study by Tiffany Denton and James Meandahl found that not only did they not improve reading performance in individuals with dyslexia, they sometimes made it worse, especially in adults. Though some participants preferred using the colored lenses to read, assessment results showed no improvement in their actual performance. So parents and educators should exert caution when deciding whether to use the colored overlays or not. Moreover, the effects that the overlays have on reading should be carefully evaluated for each individual. If a person prefers to use them, even though they don't improve their reading, that's fine, as it might increase their motivation to read. Just keep in mind that a preference for using colored lenses doesn't mean they are effective as an intervention. I'm hoping that you now have a better understanding of dyslexia, including some of its signs and symptoms, possible causes, and intervention strategies. Remember that individuals with dyslexia can lead very normal, productive lives. Their chances of success, however, are highly dependent upon receiving the interventions they need at an early age.